Good day, I'm Cheldyne Reese Pepito. My friends usually call me Che. I'm from grade 10 Zeus. 15 years old, I am born with a universal blood or so-called blood type O. I love to write stories. My favorite color is black. These are the topic I've chosen: DNA and RNA, mutation and chromosomal disorder. In this presentation, I have two objectives, and those are identify and differentiate what DNA and RNA is, and identify the disorders caused by mutations. DNA also means deoxyribonucleic acid. It is a double helix structure that is found in the nucleus. DNA also has the nitrogenous bases adenine and thymine, cytosine, and guanine with deoxyribose sugar and was discovered last April 25, 1953. DNA, also known as deoxyribonucleic acid, is a double helix that contains all the information in your life. It is located at the nucleus of the cell. DNA was discovered on April 25, 1953. It was James Watson and Francis Crick who announced its discovery. These are the following facts about DNA that you should know. DNA is your body's textbook or blueprint of life, and the carrier of all your genetic information. DNA is unique to every individual, and DNA is inherited or passed from parent to offspring. DNA has three components that are sugar and phosphate, and nitrogenous bases, where in nitrogenous bases we have adenine and thymine, guanine, and cytosine. The 70th anniversary commemorates 25th of April 1953, the day scientists James Watson and Francis Crick formally announced their discovery of the deoxyribonucleic acid or DNA that is the life's most basic genetic code. Moving forward with RNA. RNA means ribonucleic acid. It is single-stranded and located at the cytoplasm. RNA has the nitrogenous bases adenine and uracil, guanine, and cytosine with ribose sugar and discovered last 1868. RNA, also known as ribonucleic acid, is a single-stranded structure that is present in all living cells that has structural similarities in DNA. It is located at the cytoplasm of the cell. RNA was discovered by Friedrich Meister. Like DNA, the following are also facts about RNA. RNA performs many functions in an organism such as coding, decoding, regulating, and expressing genes. About 5% of the weight of a human cell is RNA. Only about 1% of a cell consists of DNA. It can also bind specific proteins or small molecules, and remarkably, RNA can catalyze chemical reactions including joining amino acids to make proteins. RNA also have three components that are sugar, phosphate, and nitrogenous bases, where in nitrogenous bases we have adenine and uracil, cytosine, and guanine. The discovery of RNA began with the discovery of nucleic acid by Friedrich Meister in 1868, who called the material nuclein since it was found in the nucleus. We have three types of RNA. We have mRNA or messenger RNA, tRNA or transfer RNA, and rRNA or ribosomal RNA. mRNA carries the genetic information for a protein from the DNA in the nucleus to the ribosomes. tRNA brings amino acid to the site of protein synthesis for assembly into a protein. rRNA is a structural and functional component of the ribosomes. Next is genetic mutation. What is genetic mutation? Genetic mutation is a sudden and abrupt changes to the genetic material of organism. It is a spontaneous change that affect the DNA or the gene or sometimes the complete chromosome. It is a change to a gene's DNA sequence to produce something different. It is to create a permanent change to that gene's DNA sequence. There are four examples of genetic mutation, that is sickle cell anemia, cystic fibrosis, albinism, and aneuploidy. But did you know there are two types of mutation? That is gene mutation and chromosomal mutation. Let's first focus on the first type of mutation, gene mutation. What is gene mutation? Gene mutation is a permanent change in the DNA sequence that makes up a gene. There are two types of gene mutation, but first let's go with point mutation. What is point mutation? Point mutation is the type of mutation in DNA or RNA wherein one single nucleotide base is deleted, added, or altered. This can lead to substitution mutation. And there are also three types of point mutation: nonsense mutation, license mutation, and silent mutation. Nonsense mutation is a change in DNA that causes a protein to terminate or end its translation earlier than expected. Missense mutation is a DNA change that results in different amino acids being encoded at a particular position in the resulting protein. And in missense mutation, there's two types: conservative missense mutation and non-conservative missense mutation, where in conservative missense mutation, the amino acid is replaced similar in function and shape to the amino acid being replaced, and in non-conservative missense mutation, it is altered in nucleotide, causes the formation of a completely different kind of amino acid in the change. Silent mutation is a form of mutation that does not cause a major change in the amino acid. And the second gene mutation is frameshift mutation. Frameshift mutation happens when the normal sequence of codons is disorganized by the insertion or deletion of one or more nitrogenous bases, given that the number of nitrogenous bases added or deleted is not a multiple of 3. The following are the diseases that occurs in frameshift mutation: sickle cell anemia, albinism, and cystic fibrosis or CF. Sickle cell anemia is a type of anemia caused by a recessive disorder through a single substitution mutation in the gene that is responsible for hemoglobin production. Albinism is an autosomal recessive disorder in which the formation of melanin is reduced or absent in skin, hair, and eyes due to the lack of activity of tyrosinase. Cystic fibrosis is a recessive inherited disorder. And the second type of mutation is chromosomal mutation. What is chromosomal mutation? Chromosomal mutation occurs at the chromosomal level, resulting in gene deletion, duplication or rearrangement, translocation, and inversion. Under chromosomal mutation, we have deletion. Deletion happens when a base is deleted from the nitrogen base sequence. Duplication occurs when a part of a chromosome is copied too many times. Inversion is when a segment of chromosome is reversed end to end. Insertion is the addition of one or more nucleotide base pairs into a DNA sequence. Translocation is the segments of two chromosomes are exchanged. Going forward with the topic aneuploidy. Aneuploidy is the change in number of chromosomes. It is the presence of an abnormal number of chromosomes in a cell. Down syndrome. Babies with Down syndrome have an extra copy of one of these chromosomes, chromosome 21. A medical term for having an extra copy of a chromosome is trisomy. Down syndrome is also referred to as trisomy 21. Klinefelter syndrome. Klinefelter syndrome is caused by an additional X chromosome. This chromosome carries extra copies of genes which may interfere with the development of the testicles and mean they produce less testosterone than usual. Turner syndrome. About half of individuals with Turner syndrome have monosomy X, which means each cell in the individual's body has only one copy of the X chromosomes instead of the usual two sex chromosomes. Turner syndrome can also occur if one of the sex chromosomes is partially missing or rearranged rather than completely absent. Cry to siya. Cry to siya or cat's cry syndrome, also known as 5p minus syndrome, is a chromosomal condition that results when a piece of chromosome 5 is missing. Infants with this condition often have high-pitched cry that sounds like a cat. And lastly, Edwards syndrome. If a baby has Edwards syndrome, they have inherited an extra copy of chromosome 18. This extra copy can be present in some or all of the baby's cell and can lead to health problems for the baby. In full forms, Edwards syndrome, the baby has inherited a complete extra copy of chromosome 18. That is all for my presentation. I hope we all learned something new. Again, I'm Cheldyne Reese Pepito, and this is my performance task in Science 10.